I made a mistake. I interrupted my career, my young career of biomedical researcher, to take care of my children and allow my wife to develop her own career. Because of that, academic instances do not allow me to continue my studies. So I don't have the possibility to have academic funds to do a postdoctoral fellow. I do not agree with this, so I try to find funds by an alternative way to develop some projects, and the first of these projects is the first stone, which one I will present to you today. The main theme of this project is epilepsy. What is epilepsy? As you can see, epilepsy is a neurologic symptom due to a transient dysfunction of the brain. It means that the neurons, the cerebral nervous cells, generate uncontrolled electrical activity in one or several regions of the brain. And the problem is that in function of the regions of the brain, you can have some different symptoms, going from just an eye blinking during a few seconds to the big tonic-clonic crisis, beginning with a tetanization of the whole body of the person who falls, followed by an uncontrolled shaking movements of all the body. So the life of all those persons affected from epilepsy, is very difficult. Epilepsy touches 500,000 people in France and roughly 50 million in all the world. And nearly the half of those persons are under 20 years old. Among those epilepsies, there is one which is of special interest for me in this project. It's temporal lobe epilepsy. Temporal lobe epilepsy represents roughly the third of all epilepsies known at that day. And it's accompanied by several phenomena nocive for the brain, like hypoxia, acidification of the medium in the regions heated by this problem of epilepsy. And among all those phenomena, there is one of special interest for this project. It's a phenomenon of pathological angiogenesis. I will explain you in the next slide what it means. The problem with TLE is that this disease is pharmacoresistant. It means that all the drugs you can give to the patients will not be really efficient. And the only efficient alternative at that day for those patients, when it's possible, is the surgical ablation of the epileptic core. So it means an intervention on the brain, a surgical intervention on the brain of the patient. So you can imagine how difficult, how dangerous this operation can be. So the objective of this project is to participate, to find new drugs which will be more efficient than the drugs we have for the moment. I told you that there was a pathologic angiogenesis accompanying the TLE, the temporal lobe epilepsy. I will try to give you just a few words concerning angiogenesis. What is angiogenesis? Angiogenesis is the growth development of new blood vessels from pre-existing vessels. As you can see in this slide, you have a pre-existing blood vessel, which wall is made of this sort of bricks that are specific cells called endothelial cells. And those cells at the region touched by an angiogenesis, these cells, which are normally tightly connected between them, will disassemble, multiply, proliferate, and then they will migrate in the environment of the blood vessel. They will reassemble next to construct, to build the new blood vessel from the pre-existing one. This angiogenesis can be done in normal physiological environment, in physiological conditions. But for example, the embryo development, you come from a small cell and you go to the baby in your arms. So in between between these two phases, you have the development of a lot of phenomena, and one of them is the angiogenesis. The problem is that angiogenesis can also occur in pathologic conditions. And this is abnormal angiogenesis. For example, in cancers, everybody knows here that solid tumors need energy, need oxygen and glucose to develop. And the problem is that a solid tumor will trigger a pathologic angiogenesis to have a lot of blood vessels to give it a maximum of energy. So I come back to the pathological angiogenesis and the TLE. The problem is that during the angiogenesis accompanying TLE, some molecules, pro-epileptic molecules, will go from the blood vessel pre-existing and pass through the endothelial cells that are disassembled in the area of the angiogenesis, and they will contribute to sustain and even increase the epileptic phenomenon. And so it's essential to block or limit this harmful angiogenesis. So the aim of the project is to identify and modulate members of two specific families of molecules that are present at the endothelial cell membranes. These two families of molecules are glutamate receptors and ASIC channels. So the molecules of interest. First of all, the glutamate receptors. Glutamate receptors are, as you can see in this slide, protein present at the membrane of the cells, and they are activated by the link of a glutamate on the receptive part of the receptor. When the glutamate is linked to this receptive part of the receptor, you have the channel part of the receptor that opens and let ions pass through him into the cell. So those ions into the cell will trigger different phenomena, molecular phenomena that will participate to modify the behavior of the cell. It's quite the same for ASIC channels, but they are not triggered by a molecule. They are triggered by the acidification of the medium surrounding the cell. And when the medium becomes acid, the channel opens and the ions pass through this channel into the cell, and here also triggers a lot of phenomena that modulate the behavior of the cell. Both glutamate receptor and ASIC channels are involved in learning and memory, and also the deleterious effect of stroke. They are also known to be involved in gliomas that are very, very aggressive cancer, brain cancer. So what are the innovative aspects of this project? Concerning TLE and other epilepsies, for the moment, at that day, few studies on glutamate receptors have been done concerning their role in blood vessels. And now, to my knowledge, as far as I know, there is no experience concerning acid channels and their role in blood vessels. So the possible contributions of this project could be a better understanding of the mechanisms of this pathological angiogenesis in temporal lobe epilepsy. Also, this project could bring new elements of therapeutic solutions for the temporal lobe epilepsy. And perhaps all this new knowledge could be extended to the other types of epilepsy. If we go further, those results could be extended to other diseases. For example, this is accompanied by a phenomenon of hypoxia. It's a lowering of oxygen to the tissues that are affected. And also diseases that are accompanied by or characterized by a pathologic angiogenesis. For example, vascular cerebral accident, and also solid tumors, as I told you before. This new knowledge could be also useful to understand better some other diseases, like neurodegenerative disease, like Parkinson's and Alzheimer. Not yet. Awesome. So, in this speech, I told you about molecules, channels, receptors. But we have to keep in mind that all this is done to try to help to cure some diseases. I could give another name to this project. This project could have the name of a young girl, aged 13 years old, I met a few months ago. The project was yet written. And this young girl is Noémie Chailloux. Noémie. And she has a TLE, and we have spoken together several times. And she has told me that her hope at that day is the surgical operation for her. And my hope is that this project doesn't come too last for her and for the others who will come after her. It's because of that I would like this project to be developed. I'm not telling you that I will find the miracle drug. I just want to tell you that those hypotheses must be tested. Because if they can bring something that could help Noémie and all the others, I think it would be wonderful for her, for the others, for us. Just to finish, we are all this here, for example, on La Coupera. Just to finish, we are all in this room. We all share a specificity, a particularity. This particularity is to have a high IQ. My way to use this particularity, to try to bring something to the others, is to do biochemical research. That's the only thing I can do with this famous high IQ. It's my way to exploit my creativity. Thank you for your attention.